Hello, I am Wachu from Baylor College of Medicine in Houston, Texas. I would like to thank Professor Natasha Ramanathan for inviting me to give this inaugural lecture. I apologize not being able to attend this exciting symposium in person. In the next hour, I would like to present to you some of the research that has been carried out in our center in Houston on cryo-electron microscopy and tomography. This slide shows different types of technologies in imaging in biomedicines. The one on the left is the familiar technology of getting high-resolution information of biological macromolecules by X-ray crystallography. The slide on the right represents another imaging modality that can provide information by the whole organisms. What I'm going to talk about this afternoon is this technique called cryo-electron tomography and cryo-electron microscopy at a resolution range between 2 angstrom all the way to 80 angstrom resolution. And in the last 10 years, cryo-electron microscopy has been advancing rapidly in terms of publications and also structured deposit in the protein data bank as well as electron microscope data bank. In the last year, we have over 3,000 3D EM density map deposit in EMDB and over 1,000 coordinate or model deposit in the protein data bank. Among all these structured deposit in either of the database, the resolution span between 2 angstrom all the way to 80 to 90 angstrom resolutions. As you see in this curve, in the last 6 years, there has been readily increasing numbers of the structure being solved by the community around the world. And in our center, back to in 2008, we have begun to solve the cryo-EM structures of a chaperone and a virus particle from which a backbone trace of individual proteins of these macromolecular assemblies can be accomplished. And in the last 6 or 7 years, we have solved more than a dozen structures of these molecular machines. Their protein backbone is possible. And all this work was done with conventional electron microscope. The images were recorded in either photographic film or CCD camera. In the last 3 years in particular, the field has had a sudden rise in resolution for cryo-EM structures solved not only in our lab, but many labs around the world as well. As noted in these figures on the distributions of structure in the atomic resolution between 5 and 2 angstroms. Particularly just last year's alone, we have close to 100 structures being solved by cryo-EM at 3 and 4 angstrom resolutions. And the question is, why all of a sudden in the last 3 years, such a large numbers of structure can be solved at these kinds of resolutions? Here are my perspectives on the advances in cryo-EM in the last few years. The first and the most important one is the availability of direct electron detectors that allow us to take images which can preserve both low and high resolution information at high data transfer capability. And I would demonstrate how this works in some of the slides later on in the talk. The second reason of such events is because the availability of image processing software and modeling software that have been developed in many labs, and those software are openly accessible to any other users around the world. Thirdly, that in the last 3 or 4 years, the community has begun to be aware of the importance of establishing rigorous criterion in validating both the map and model before they are published and before they are deposited to the EMDB and protein data banks. Such establishment allow the community to complete their structures with high reliability and accuracy. And the fourth reason on the recent advances is because there are many molecular machines which have been ready for Cryo-EM, and those materials are commonly found in crystallography lab who will not be able to crystallize those molecular machines but are quite ready for Cryo-EM study at high resolutions. So with these 4 reasons, the Cryo-EM has become a very popular technique among the structural biologists to pursue near atomic or atomic resolutions, structural determinations. Now, in our lab, in the last couple of years, we have the availability of these direct electron detectors. We have 3 types of detectors from 2 different commercial vendors, and we actually have solved several of these structures, including some of those which were specimens that we did in previous years. And in our experience with this new direct electron detector, we were able to improve our resolution of some of the old specimens by 1 to 2 angstroms. These are relatively significant because these near atomic resolutions around 4 or 5 angstroms, if we can improve 1 or 2 angstroms, it would allow us to not only trace the backbone, but also to resolve the side chain density in the protein molecule as well. And so today I'm going to show you examples how we use these kinds of data to retrieve the near atomic resolution structures. Here is a pipeline of the whole data acquisitions and data processing steps. And first, we have to purify the material similar to the crystallography studies. Our aim is to prepare the specimen not only functionally pure, but conformationally homogeneous and also rigid in terms of conformations. The difference between the Cryo-EM and other biophysical techniques is the requirement of the sample concentration and the amount. Generally speaking, we need a specimen concentration between 0.1 to 1 milligram per ml. The total amount of the studies can be as little as 100 microliters. Once those specimens can be prepared biochemically in a wet lab, the first step in the Cryo-EM procedure is to freeze it in a very thin layers of vitreous ice. By vitreous ice means ice in an amorphous form. And each of these preparations for the Cryo-EM study requires about 3 to 5 microliter. So as a result, the specimen requirement is much more relaxed compared to crystallography or NMR studies. And once those frozen specimens are prepared, we put a electron microscope, and most of the microscope, either the old one or new one, are quite readily usable for recording good quality images. And as a result of those data collections, one would have a stack of images. Each image represents a 2D projection of the single particle in a matrix of vitreous ice. Like for example, in this particular image, this dark color circle corresponds to a projection image of a single particle embedded in surrounding matrix of vitreous ice. And from here, with this stack of images, the image processing begins to combine each of these particle images to generate its three-dimensional density maps. Now, in these steps of data processing, it could be pretty tedious, but it could be pretty straightforward, depends on the particular types of samples. And the major thrust in the image processing is to find, we call, orientations of each of these particle images, and then make all the image corrections due to artifact generated in the electron images, and then combine them coherently to generate a three-dimensional density map. When the map resolution is good enough, then one can build individual model for each of the protein components, and eventually each of the protein components models are built and combined to generate an atomic model before they are deposited into a protein data bank for the model and the EM data bank for the density map. In the entire pipeline of this Cryo-EM protocol, the limiting steps can be at this step in preparing good specimen, or it can be in the second steps to have the right thickness of the eyes, and also the particle has to be of different random orientations. In terms of data collections, it's pretty straightforward, and in terms of data processing, depends on the structural heterogeneity of the sample. If the structural is very homogeneous from particle to particle, the data processing can be straightforward. But if the structures are heterogeneous among particles, or if the structures of the particles are full of dynamics in terms of different conformations, the mergings of data can be challenging. So, like any other projects, the challenge of these, as I point out, is to be able to purify the material in good quantity and quality, as I described just a while ago. Here is an example of the specimen. I like to spend a few minutes with you how we collect the data, process the data, and interpret the data. This is a double-strand DNA bacteriophage. This diagram shows the assembly process of this protein to form a infectious particle from an initial assembly of the cold protein with the scaffolding proteins, and with additions of one of the protein apparatus through which the DNA is encapsulated. At the same time, the scaffolding protein will be exit out the capsid particles, and when the DNA is fully encapsulated, and then the tail fiber will be attached to this special vertex in an infectious particle, this will become infectious to a bacterial cell, and then the genome will be released in the cell in such a way that new virus particle will be synthesized in the cell. So in the remaining of my talk, I will first talk about how we get a structure of this infectious particle, and secondly, we'll talk about the so-called procapsid before the DNA is encapsulated, and then lastly, we'll talk about how we can determine the structures of the phage particle inside the infected bacteria. And here is a slide to show the image of the biochemically purified bacteriophage called P22. We use one type of direct electron detector called D20. And one of the usefulness of such a detector is very high frame rate. This means that in each of the specimen area, we can take multiple frames, and then with all the frames, we can do computational manipulations to realign the frames so that each frame is exactly aligned to each other. Here I'm going to show you how different numbers of frames are being aligned each other. So at the beginning, I have only, like here, I stop here, it's only 5 frames being put together. The contrast of the particle is relatively low. Here are the surrounding ice. And then if I add more frames, and then the particle contrast become better and better. Now, why is it important of having this capability align different frames? Because when the electron hit the specimen have a tendency to move around under the electron beams. But after the post-data acquisition with alignment from frame after each other frame, and we can put all the frames in register in such a way that the image can be much sharper than just purely sum all the frame without registering it. And so if we continue to have more frames after its alignment, it becomes much and much better contrast. Like for example, these up to 23 frames, each of these virus particle of P22 become much easy to recognize, and you can actually see different substructural features like the fingerprint feature, which is a characteristic pattern of the DNA seen in 2D projections. And the diameter of this P22 particle is around 700 angstroms. So with these types of high contrast image, it allows us to determine, we call orientation of each of these particle much more readily. And by orientations, I mean the centers of the particle and also the Euler angle of the particle. By Euler angle, there are 3 angles per particle, which represent the coordinates of the particular orientation with respect to a common frames of reference of all the particles. And so we normally use tens of thousands these types of particles to do the reconstructions. Before we do the reconstruction, this slide to show you how we evaluate the quality of those images. And this, the one on the left in half of the pattern, represent the power spectrums of the particle images from one specimen area. And this oscillating contrast between black and white is the characteristics of any electron images, which due to the defocus setting of the image was taken. And so when we change the defocus, the oscillating patterns will vary. And the one on the left is the simulated power spectrums of a specific defocus value. As you can see, if we only sum all the image frame without careful and computational realignment, some of the high resolution information is lost. But the pattern on the right represents the particle image has been realigned in such a way that all frames are in register for each the particles. So you can see this oscillating pattern as a characteristic. The defocus of these particular images show up at much higher resolutions than that with only a simple sum of the frames. And so this pattern shows the power of these direct electron detectors allow us to retrieve at the onset before we do any image reconstructions to restore the high resolution information for each of the particle images. And so here is the data processing steps, and we have to generate 2 data sets for the entire data set. For example, in this particular example of P22 phase, we have 22,000 particles, and we split the total numbers of particle image in 2 data sets, and each of them has 11,000 particles. And for each of these 11,000 particles, we gradually improve its reconstruction resolution from initially just a sphere and gradually to see structural features, and finally when it gets to high resolution we see much structural details. So we did first in half of the data, and then we did secondly in the second half of the data. So as a result of that, we have 2 3D density maps from each half of the entire data set. The reason we generate these 2 independent density maps is because we want to make an estimate of how similar the structures are between these 2 independently generated density maps. And so with these 2 density maps, we actually compute this structural similarity with a parameter called Fourier cell correlation, which is a dot product of the Fourier transform of the 3D density map of 1/2 of the data with the complex Fourier transform of the density map of the second half of the data, and normalized by the dot product of the both Fourier transforms. And so this is a part of these correlation functions as a functions of frequency. At low frequency, means low resolution, both density map agree very well, close to 1. As the resolution get higher, the 2 density map become less similar, as shown in this fall off phenomenon of these Fourier cell correlations. It has been analytically demonstrated by Richard Henderson that if we use the value of 0.143 for this Fourier cell correlation, it would be a good representative of the resolutions of the entire data set to generate a final map. And so in this particular example, we have a 3.3 angstrom with 22,000 particle images of P22. And here is a slide to show what you can see at this 3.3 angstrom density map of P22. This is a surface rendering display of the density map, and as shown in this static image, it has the characteristic pattern of this hexameric we call capsomere, and there are 60 of these capsomere, and then 12 of these pentameric appearance capsomere, and all together 1 asymmetric unit we define as 1 hexameric capsomere and 1 subunit of this pentameric capsomere. So this particular movie, what I'm going to show you is to zoom in 1 of these asymmetric unit with 1 copy of hexamer and 1 subunit of the pentamer. And the density of this map is good enough that allow us to delineate the molecular boundary of individual protein subunit, which we represent with different color, like this orange subunit have density sticking on top of the adjacent protein in red, and then when you rock this density toward the left, part of the other density of the orange subunit is tucked in the bottom part of the adjacent green subunit. So now in delineating the boundary of these kinds of subunit can sometimes be challenging, but it is a iterative process. And here is a computational extractions of 1 of the subunits. As you can see, the continuity density is quite good. In such a way, we can actually trace the continuity of the density from 1 end of the molecule, like the N termini of the molecule, to the C termini. So this particular blue curve, color curve, show the topology of these polypeptide, namely the tertiary structures or the backbone trace of these polypeptides. And as you can, like any other protein, the topologies of the polypeptide can go from the left to right, top to the bottom, and back to the top again. And so as a result, the entire polypeptide with over 430 amino acids can be traced entirely. And so now with this topology trace, it can be done by software company, like for example, the Pathwalker developed by Matt Baker in our lab can do this trace automatically. Alternatively, we can actually use any other existing crystallographic software to make such a trace. Now, having obtaining such a trace, our next step is to build a true full atom models based on the density. And we use the crystallographic software called Phoenix. Then we can optimize these trace. By optimizing, I mean to assign the side chain density to our map, and according to the amino acid sequence of this protein. And number two is to refine the stereochemistry, namely the connectivity between the chemical bonds of every atom. This movie shows you how we do in this process, which is a iterative process to fit globally as well as locally to the density. Like for example, there are densities which can be used to fit and to build the presence of the side chain along the entire polypeptide. So both these positioning of each the atom of the side chain as well as the backbone are done iteratively. And all this can be done with this software called Phoenix, which is a very popular software in crystallography. And so once this entire full atom is built for one polypeptide, we can, in this movie, to look at how could it fit between the model and then the actual density. What I am showing you here are the experimental density of all the aromatic residue in these polypeptides when these are sewn together with the entire polypeptide backbone. So the Y diagram is the density in our experimental map with all the aromatic residues. As you can see, the model built with this density actually looks really well in terms of the rotamer conformation of each of the side chain with the density map. And such a nice match between the model and density is seen actually throughout the entire polypeptide. So having done this, we can then in this slide to show you not only the aromatic residue, but all the residue in these 430 amino acid long polypeptides for one protein subunit. And so the one on the left is this long alpha helix, the one on the top is the short helix, and the one on the right are the two different loops area, and the one on the bottom shows the four anti-parallel strand density of these single polypeptide subunits. As you can see, almost all the side chain model match well with the density, except the negatively charged residue like the glutamic residue as well as the aspartic residues. And so this actually turned out, it's interestingly, that all the negatively charged residue density are either absence or of very low density value in our Cryo-EM map, as well as in any other map that structure has been solved by Cryo-EM. The reasoning of such a low density appearance of these negatively charged residue can be either caused by the radiation sensitivity in terms of damage of this negatively charged residue, or a scattering difference between negatively charged residue than the neutral and positive charged residue by the electron, which carry a negative charge. and so the exact reasoning is yet to be determined. And so now, having built the structures of one polypeptide, we can begin to build the model for all the other 6 polypeptides in one asymmetric unit. Just to reiterate, there are 60 asymmetric units in this virus particle, and each asymmetric unit has 7 polypeptides. Now, even though all the polypeptides are chemically identical that make up this P22 phage, due to its geographical locations, the structure of each of these 7 polypeptides do not have to be identical. So as a result, we build the model for each of these 7 polypeptides. When we superimpose the full atom models, is clearly that they are not identical. Like, for example, the N-termini are rather diverse among these 7 proteins, and as well as this long loop area. And if we measure quantitatively speaking, in these N-termini, they are very much deviate among the 7 subunits. And interesting enough, there is one small helix which are facing to this center of these exon or pentons, also have a large variations, which I will explain later on why that may be the case. So having built the 7 subunits model, then we can reassemble all this model to build the full atom model for the entire virus particle, which has 60 asymmetric units. So now, having done one structure of the material virion, I'd like to switch gear to show how we approach to do another structure we call a Procapsid, which the images are shown on the left. In the Procapsid, the diameter is about 100 angstrom smaller than the material virion. And secondly, the features of these Procapsids look like to be more roundish than the material virion, look like more angularish. And then, in fact, there are more features like the fingerprint, as I point out in the movie, which the fingerprint disappear because there's actually no DNA in the Procapsid. So using a similar method I described for the material virion, we actually solve the structures of the Procapsid at 3.8 angstrom, slightly lower resolution than the material phage, which I talked about a while ago. And so now, one of the interesting aspects in solving the Procapsid structures is to try to find out how this reduction in size of the phage particle can occur, or in other words, how the expansion from a Procapsid to the material virion will take place. And so, in order to do that, we also build the individual model of each of the subunit on the Procapsid. The one on the left is the protein fold of the co-protein of the Procapsid. The one right is the one that I talked about a while ago. As shown in these two slides, it's quite obvious the N-termini undergo a large conformational change, become a kind of flexible loop in the material virion, where in the Procapsid it's actually form a sort of a helix, and then also the extensions of these long loops are also different. And there are also variable parts of these, like for example, these helix here would also have a different conformation in the Procapsid structures. So in the next slide, I would put both of these structures together to give you a sense how this conformational variant actually is taking place. So here is a movie which I morph two asymmetric units First, these are the asymmetric units of the procapsid without the DNA. As obviously, in these structures, there's a big opening, and they also seem to be having a loose contact between adjacent subunit. Now, if I morph these structures from the procapsid to the mature virion, you see there are actually quite a bit of conformational differences. And in the mature virion, this big hole seems to close up. If I morph it again, you will see that actually this loop area, which undergo a large conformation, just like closing up the hole. And in the mature virion, there's actually much closer contact between the adjacent subunit than the procapsid. And the loop area also undergo quite a bit of change, as well as these N-termini that kind of destabilize the helix, become a floppy loop. And now if we look at these asymmetric units in a side view, and you can see actually the thickness of this capsid shell is reduced when it transforms from the procapsid to the mature virion. Again, this is very characteristic. It's from this helix to loop when you mature from procapsid to mature virion. So the interesting aspect about this is during this capsid particle transforming from a procapsid to the mature capsid with the encapsulation of the DNA, there is a lot of conformational changes of the protein capsid in such a way the whole goes up, and then subunit interaction also get modified. The thickness of the capsid also get thinner, and in such a way to allow the DNA to be encapsulated. So I think this shows about the usefulness of the Cryo-EM. You can do a structure not only in one functional state of a molecular assembly, but also in multiple functional states, like in this case, before the DNA is encapsulated and after the DNA is encapsulated, we begin to understand the structural mechanisms of making the phage particle go from one size to a bigger size upon the DNA entry. And so now, so far, what I have shown you are the atomic resolutions of the procapsid and the mature virion. As shown in this diagram, they're supposed to have some other scaffolding protein in the procapsid. So we then want to show you where they are inside. So far, I only show you only the capsid protein, but not yet all the other proteins. And so here is now shown you by looking at inside of the procapsid. The red density proteins are the density that belong to the scaffolding proteins. That we know that because we trace all the density of the capsid proteins with all the amino acid. Whatever left that cannot be accounted by the capsid protein has to be the scaffolding protein. Interesting enough, the ratios of the scaffolding protein and the capsid protein is 1:1. And so, the detailed analysis of the interactions of the scaffolding protein and the capsid protein can now be examined in detail. I just would not have time to show you all the detail, but the information is definitely there that we can mine from the structures. Now, so far, I have talked only about the capsid protein. You may remember all the phages actually in one of the 12 vertices, there are additional protein apparatus through which the DNA is encapsulated. So in these particular structures, we actually relax all the symmetry in deriving the density map. As a result, our resolution on these structures is lower, only at 7 angstrom for this mature phage. And then in this case, we see not only the capsid protein, but also the other proteins in the in the special vertex. And here is a slice of those density. You can see all these ring structures correspond to the average density arrangement of the double-strand DNA inside the virus particle. Here are a lot of proteins making up this special vertex. And in order to make the long story short, I only summarize what we can interpret in this asymmetric reconstructed density map. We have the GP5, GP1, GP4, GP10, GP9, GP26 that make up this special vertex, and each of these protein represent different color and actually have different numbers of copy. Like this portal protein, the green protein, there are 12 copies, which are surrounded by 10 copy of the capsid protein that we talk about in detail in previous slide. And so from these kinds of density map, even at 7 angstrom, we're able to delineate exactly what types of interactions are involved with each of the 12 copies of portal protein with this surrounding 10 copy of the capsid protein. So I don't have time to go through all the detail about the interactions, but even at 7 angstrom, we actually learn a lot about the molecular interactions that make up this particular protein apparatus through which the DNA is encapsulated, and also when this mature virion is made. This called needle is the density that involved in the interaction with the cell surface when the phage are infecting the bacteria host. And so now I have gone through the structural determinants of Procapsid, also mature virion, not only in high resolution detail of the capsid protein, but also the subnanometer resolution, quick description of this special vertex. In the next few slides, what I want to share with you is what happened to the structures when the phages are newly synthesized in the bacterial cell. So here is an example of seeing this entire bacterial cell in a electron microscope, and this particular bacteria is called Synococcus, which is a marine bacteria, and the phage we infect is not P22, but it's a marine phage called CEM3DIP. And when we look at it in the electron microscope, usually the specimen is put around these positions, but then we have a new special optical phase plate, which is a device which is made up of a thin carbon film about 250 angstrom in thickness at the focal plane of the objective lens, with a hole in the middle. And you can see that when we image these bacteria with the phase space, that actually quite substantial detail can be visualized, whereas when there's no phase space, it's really hard to delineate the subcellular components. So the physics behind this is similar to the Zernike optics, which the scatter wave and the unscattered wave of the electron have a phase shift by 90 degrees that responsible for such a contrast enhancement. And the thickness of this particular cyanobacteria is around 0.7 micron, which is small enough for the electron beam to penetrate through and form images. And so now, since no two bacterial cell is structurally identical, we do the 3D reconstruction one cell at a time. And here is a tomogram of the cyanobacteria infected by the cyanophages. And then we do a reconstruction of one tomogram, which is equivalent to a CT scan of a human body. So by taking different two specimen images, we can combine it to generate a 3D tomogram. And here what I'm showing you is a slice by slice of this 3D tomogram of these cyanobacteria infected by cyanophages. In this particular case, the infecting cyanophages has the typical structural arrangement as P22, except that it has an additional density at the opposite vertex of the face. We call the horn density sticking out at the opposite end. And so here are a tomogram of the cyanobacteria, which has been infected by the cyanophages for a little bit long time, which represent the kind of late infecting stage of the bacteria. And as you can see, there's a lot of features on these ice-embedded cyanobacteria infected by the cyanophages without fixative, without negative stand. And so in order to see the feature further inside the cell, what I am going to do is to segment out some of the familiar subcellular feature as well as the infecting and newly synthesized phages in this tomogram. And as annotated in the next few script of this movie, now I show you here is the cell wall of this cyanobacteria, and then parallel to cell wall, this is a thylakoid membrane because this is a photosynthetic bacteria, and then within this the bacteria there's about 1,000 angstrom wide diameter particle we call carboxysome, which contain a lot of rubisco, which is an enzyme to fix the carbon dioxide in the photosynthetic reactions. But all these rubisco form within a particle called carboxysome. And in addition to that, there are plenty of ribosome in this bacteria like any other cell. And we see one vesicle in outside. And then we see in these red colors we call infecting phages are the phages at the close vicinity of the bacterial cell surface. And inside the cytosol of this bacteria, there's a lot of purple color particle which has the size equivalent to this syn5 bacterial phage. Now I am now get rid of the density and showing you these only the annotated features. Of course, there are many other subcellular features we are not annotated in here. Now, interesting enough, when we look at these particular phages, that we interpret these as these phages are in the act of transfer a genome from the phage to the cell. Interesting enough, we can see the density right here is penetrating through the outer envelope of this bacterial cell, equivalent to this phage tail, and then this horn at the opposite end actually is pointing away from the bacterial surface. That suggests to us the function of the horn probably has nothing to do with the infection process. And then when we pan to the other end of these bacteria, because this particular bacteria is at a late stage of infection, so it is actually as part of the cell begin to be lysed, and some of these appear to be the polyribosomes that are kind of leaking out from the cell to the extracellular part of the medium. And so this is what we learned in a single tomogram. There are actually a lot of subcellular informations that can be mined. Now, what I want to focus in the next couple of slides is to look at some of these newly synthesized phages, what happened at different infection time course. So here are 4 different examples of these cyanobacteria, which has different amounts of phages inside the cytosol. Like, for example, this is the one had lots of bacterial phage inside the cytosol. We consider the late stage of the infection. And here we call intermediate stage because we only see a few newly synthesized phage. And then we call early infection because we only see phages outside but none inside at all, and this is an uninfected cell. So with this kind of tomogram, we took a lot of them with these Zernike phase contrast optics, and then the contrast good enough that we can identify each of these cell subcellular components, which are summarized in this slide. And so here are these individual subbottoms of this tomogram, which when we measure the size and then the features, we align and average. Some of these particles look alike, and we get an average sum. Like in this case, there are 96 of these subbottom density look like a mature phage, like with this horn density as well as a tail. And here is a slice, cutaway view of this virus particle. And then the second types of particle we're able to see is the horn is missing, and the third one is the tail is also missing, and the fourth one is that something nothing is inside the particles except there's a density in one of the vertices, and then also the diameter is the same as the mature or fully DNA encapsulated particle. And the last types of particle we are able to see is look like roundish looking, which interpret as the Procapsid, analogous to the P20 case I talked a while ago. Now, interesting enough, based on these types of analysis, we make a couple of conclusions. First, we conclude that this horn density probably is the last components of the protein assembled in the phages. And then, because we have not seen particle with only the horn without the tail. And the second thing we learned from these kinds of analysis is the expansions of the diameter of the phage particle, even though not a lot of DNA density are found inside. This suggests that the expansion does occur at the very early stage when the DNA begins to encapsulate. And then the third observation we made is this particular density in one of the trial vertices, which we interpret as the terminase. It has been shown biochemically when the DNA is encapsulated in the terminase is required to generate the ATP energy for the encapsulations of the DNA. And the last point I want to mention is that the number of particles found in all these tomograms appear to be different. Interesting enough, this particular case, which has expanded configuration but no DNA, as only a very few particles are found in our data set. This suggests to us that this particular class of particle probably is very transient. That means they don't last a long time in the cell. That means the transitional period from this empty-looking expanded particle would transform into the DNA encapsulations in a very rapid rate. So based on these types of analysis, we suggest an assembly pathway of this phage particle, initially form a procapsid with the help of the scaffolding protein, and subsequently expand upon entries of the DNA, trigger a lot of conformational variations, and then also the scaffolding the protein escaped through the hexon with the opening of that. And then finally, when the DNA is encapsulated, there is a substantial change also with additional tail density attached to this vertex, and finally the horn is being assembled, and that's the last steps. So I think this set of tomographic analysis really reveal this assembly process of this phage particle inside the cell. Currently, the structural determinations of these are rather low, around 40 to 50 angstroms. I think the prospect of extending to high resolution is very, very high. And the requirement is that we need to collect more data and expand the data set so we can do the averaging with more particles to enhance the statistic in order to put the resolution to a better level. And finally, I'd like to summarize that through my presentation, I demonstrate using this P22 phage and P20, and then also the cyanobacterial phage with the cyanobacteria to demonstrate how we can use the single particle cryo-EM to determine the atomic resolution structures of biochemically purified phage, so that we can visualize the entire polypeptide backbone as well as the side chains. And we also show that the cryo-EM is a technique that can reveal structure not only in one functional state, but multiple functional states, like in the Procapsid as well as the mature variant of P22. In the last part of my talk, I demonstrate how we can use the electron tomography to look at the biological process, in this case, the assembly of the phages inside the bacteria. And I think this last part of my talk to me is truly exciting area of future research in cell structure biology. And finally, I'd like to thank all the colleagues that make this work possible. Corey Rick, who is a graduate student, developed the model building process from a cryo-EM density map obtained by Donghua Chen, currently at Stanford University. And then Zhao Wang and Joanita Jacana help the data collection of the P22 project. The tomography project was spearheaded by Wei Dai, who's currently at Rutgers University, and assisted by Ted Kang and Caroline Fu for the data collections. And Dr. Liu, Pintari, Smith, and Lucky has contributed the development of the software for image processing, as well as the model building. And the project I described to you for the P22 and the cyanobacteria infected with cyanophages are in collaboration with Jonathan King at MIT and Wen Zheng, who's a former member of our lab now at Purdue University, developed also substantial amounts of software for the reconstruction of the bacterial phages, and in collaborating with Paul Adam, who is a protein crystallographer, we developed the software for the model building of the cryo-EM map, and our work is being supported by NIH and the Wells Foundation. I thank you very much for your attention, and I hope that I have communicated my excitement on the cryo-EM, and I hope that all of you will practice cryo-EM in the future. Thank you very much.